Good morning. I'd like to thank the organizers for giving me the opportunity to present my PhD results obtained so far. So this work deals with the regulation of autophagy by chemotactic GPCRs and its potential involvement in glioma cell invasion. Now, chemotactic cell migration is a key aspect of many physiological processes, such as embryonic development, wound healing, or immune surveillance. But it also takes part in a few pathological processes, such as tumor progression. You can see in this video, for example, a brain tumor stem cell escaping the mass and migrating through a microfluidic system. So let me start by introducing the different steps involved in chemotactic migration. In response to a chemotactic stimulus, the first step is the polarization of the cell and the formation of the front end of a structure called the lamellipodia. The lamellipodia is stabilized by forming integrin-mediated adhesions with the extracellular matrix, and this allows the cell to contract and translate the cell body forward. And this is then followed by de-adhesion at the rear end. Now, to form these new adhesions, the cell will in part have to recycle integrins from its rear end via recycling endosomes. And to launch this whole mechanism, most of the chemotactic stimulus bind to cell surface receptors that belong to the G-protein coupled receptor family, or GPCR. Now, as a prototypical chemokine receptor, the GPCR named CXCR4 has received much attention. Together with its ligand, the stroma cell-derived factor, or SDF1, they have been shown to be involved in the migration of leukocytes or endothelial cells, but also in cancer cell invasion. Now, besides these classical chemokine receptors, other GPCRs also display chemotactic activity. This is the case for the receptors for some vasoactive peptides, such as urothionein 2, or U2, which by binding to its receptor, UT, has been involved in the migration of fibroblasts or endothelial progenitors. Now, interestingly, our team has recently shown that UT is also involved in cancer cell invasion, as was the case for CXCR4. And our team has recently shown similar expression profiles of these two GPCRs and their ligands in glioma explants from patients. So one of the main objectives of our team is to try to understand the intracellular signaling pathways that are activated by these GPCRs. And the object of my PhD was to investigate the involvement of autophagy. Now, autophagy is a lysosomal pathway that is involved in the degradation of long-lived proteins or organelles, and it takes part in a few cellular processes such as differentiation or development, and of course, cell migration. A number of studies have shown a role of autophagy in cell migration, but often with conflicting views. On one hand, it has been shown that key proteins that are involved in migration, such as integrins, can be degraded by autophagy, suggesting that autophagy may inhibit chemotactic migration. On the other side, it has been shown that inhibiting autophagy blocks the cell's ability to migrate, suggesting that autophagy may induce chemotactic migration. So these two seemingly opposing views beg the question, is autophagy an inhibitor or an inducer of chemotactic migration? Now, as a reminder, the autophagic process starts by the formation of a multi-membrane structure called a phagophore, which will elongate, capture cytoplasmic material, and close off to form a mature autophagosome. The autophagosomes then fuse with lysosomes to form the autolysosome, where its interior is degraded and used for the creation of new proteins. Now, to this day, there has been no clear link established between chemotactic GPCRs and the autophagic activity. The object of this study was therefore to understand the impact of chemotactic GPCR activation on autophagy and understand how this modulation of autophagy could affect chemotactic migration. And to do this, we used two cell lines, the X cells overexpressing either CXCR4 or UT, and the U87 glioblastoma cell line that naturally expresses both GPCRs. So we first assessed autophagic activity using a fluorescent probe called CytoID, which can bind directly to autophagosomes. The fluorescence intensity emitted by each cell is therefore proportional to the quantity of autophagosomes it contains. So cells were expressing UT or CX4 were treated with their respective ligands, and the fluorescence intensity was measured by flow cytometry. As you can see, treatment of cells with SDF1 and U2 reduced the amount of autophagosomes per cell. But as you can imagine, the amount of autophagosomes is at a balance between the speed at which they're formed and the speed at which they're degraded. So to test the activation of these GPCRs on autophagosome biogenesis, we chose to inhibit the degradation with chloroquine, a drug which inhibits lysosome acidification and therefore inhibits autophagosome degradation. So as you can expect, when we treated cells with chloroquine, we caused the autophagosomes to accumulate in the cell. But as you can see, the inhibitory effects of both GPCRs are still present, suggesting that the chemotactic GPCR activation inhibits autophagosome biogenesis. This brought us to focus on the different steps involved in autophagosome biogenesis. A number of proteins are involved by being sequentially recruited to the phagophore membrane, the first of which is mTOR, which constantly inhibits the formation of new autophagosomes. When we lift mTOR inhibition, we have activation of a PI3 type class 3 complex, which forms PI3P at the membrane of the phagophore. And this serves as a recruitment platform for a protein called WIPI, which then recruits this ternary complex and then the LC3 protein. So we looked first for a potential effect of chemotactic GPCR activation on these early steps of autophagosome biogenesis, but found no significant effect. This brought us to focus on the LC3 protein, which I mentioned is the last protein to be recruited and the only one to stay at the surface of a mature autophagosome. We used the green fluorescent LC3, and when you stimulate autophagy, you have the labeling that goes from diffuse to a more punctate labeling, the dots corresponding to autophagosomes, and that can be counted. So we treated cells with an mTOR inhibitor to stimulate autophagy. As you can see, we have the appearance of this punctate labeling. When we treat cells with either ligand, we see that chemotactic GPCR activation reduced the number of LC3 dots, suggesting that they reduced LC3 recruitment to the phagophore membrane. So as I mentioned, we had no effect on these early steps, but LC3 recruitment was strongly inhibited. This brought us naturally to focus on this complex to understand how it may be affected by chemotactic GPCR activation. Now, earlier studies had shown that the proteins that are part of this complex, HEG5 and HEG16, are recruited to the plasma membrane. There they take part in the endocytic formation of pre-autophagic vesicles, which feed the phagophore with an important source of phospholipids. This seems to be very important for the formation of those autophagosomes, since when we inhibit this step using dinosaur, a drug which inhibits the scission of these vesicles from the membrane, we also completely block the formation of mature autophagosomes. So we wondered if the chemotactic GPCRs could affect the recruitment of these two proteins to the plasma membrane. We first looked at HEG16. So once again, we treated cells with dinosaur to cause their accumulation at the plasma membrane, as you can see by this punctate labeling. But when we treated cells with either SDF1 or U2, you see that we reduced the number of these pre-autophagic vesicles, suggesting that HEG16 recruitment to the plasma membrane was strongly inhibited. But as we know from the literature, the membrane binding site in this complex is held by the HEG5 protein. So we next look how HEG5 could be affected. As we noticed, a strong portion of HEG5 localizes to the plasma membrane already. And treatment of cells with either SDF1 or U2 once again strongly inhibited recruitment of HEG5 to the plasma membrane. So the question was, how might HEG5 be affected by these chemotactic GPCRs? Now, interestingly, earlier studies had shown that HEG5 could be the target of a protease called calpain, and cleavage of HEG5 by this calpain gives rise to a smaller fragment that can no longer take part in the formation of autophagosomes. So to test this theory, the involvement of calpains in our models, we treated cells with a calpain inhibitor, ALLN, and once again we looked at the recruitment of HEG16 to the plasma membrane or LC3 to the membrane of autophagosomes. Like as before, we had a reduction of both recruitments after chemotactic GPCR activation. But as you can see, treatment of cells with ALLN in both cases re-established recruitment of HEG16 to the plasma membrane and recruitment of LC3 to mature autophagosomes, suggesting that the effects noticed till here might be relayed by calpains. To go a step further, we wondered if we might inhibit these effects by overexpressing HEG5, possibly compensating its cleavage. So once again, when we overexpressed HEG5 in our model, we were able to block the anti-autophagic effects of the chemotactic GPCRs. So to put these results back into context of chemotactic migration, we wanted to know how this modulation of autophagy might affect a cell's ability to migrate. To assess migration, we use a transwell Boyden chamber, in which cells are placed in a top chamber, separated from a bottom chamber by a porous membrane, and in the bottom chamber, we place the chemokine. This creates a gradient which drives the migration of the cells to the other side. And by counting the number of cells that have crossed them, we can assess their ability to migrate and to respond to a gradient. Now, as expected from two chemokines, U2 and SDF1 stimulated the migration of our cells. But as you can see, this seems to be dependent on calpain activation, since the calpain inhibitor in both cases inhibited the pro-migratory effects. And this was also the case when we used siRNA silencing of the calpains. What's even more interesting is that the simple overexpression of ATG5 gave the same results, where we were able to block the pro-migratory effects of both chemokines. And we obtained similar results in the UID7 glioblastoma cell line. So the final question we asked was what might be the link between autophagy and cell migration? So as I mentioned earlier, to form new adhesions, the cell has to recycle part of its integrins via a recycling endosome. But it has been shown that autophagosomes can interfere with this process by fusing directly with the recycling endosomes and bringing the integrins to degradation. So this brought us to look at the formation of these new adhesions. So we'll use the vinculin immunolabeling, vinculin which is part of these complexes. And what you can see is that when you treat the cells with U2, you have the formation of the laminipodia and adhesion complexes at the front end, which we can count also. But simply overexpressing ATG5 once again inhibited the formation of these new adhesions. What's even more interesting is that it seems that ATG5 is important in this process, since silencing it partially reproduced the effects we observed with the chemotactic GPCRs. So these results suggest that the inhibition of autophagy is necessary for the formation of adhesions. So this is the model we have right now. We believe that chemotactic GPCR activation will cause a calpain-independent cleavage of ATG5, which inhibits the formation of these pre-autophagic vesicles and therefore the formation of mature autophagosomes. And that this inhibition of autophagy might be important for the recycling of integrins to the laminipodia. In the future, it might be interesting to see if other autophagy proteins may be affected by chemotactic GPCRs. And we might also perform in vivo studies to see if by modeling the levels of ATG5 or calpains in glioma cells, we will also be able to modulate their invasive properties. Thank you for your attention.
You said that ATG5 overexpression stimulates the migration.
It seemed to inhibit the pro-migratory effects of the chemotactic GPCRs when we overexpressed ATG5.
Okay, yes, that's what I thought it was only overexpression. I didn't know. So it is in the presence of the chemokine.
Yes, it's also in presence. Okay, we didn't notice any effect of the simple overexpression, but when we added the chemokines, that's when we noticed that the cells were no longer able to, we had no pro-migratory effects.
Okay, and would you say that U2 is a better chemokine than SDF1?
On the X cells, it seemed to be the case. We had better effects of U2, but when we performed the same experiments on the UID7 glioma cell line, SDF1 seemed to be more effective. But this might also be due to expression levels, since we overexpressed both GPCRs in X cells. Yes.
You hypothesize that SDF1 as well as U2 would be involved in migration of tumoral cells. And you have chosen a model of two tumoral cell lines. So I was wondering, why did you overexpressed U2 and CXCR4 receptors?
Yes. So in X cells we noticed did not naturally express UT and very little CXCR4. That's why we chose to start the experiments in these cells where we could control the levels of these receptors. And then we went to another cell line, the UID7, which in this case naturally expressed at strong levels the two receptors.
Yes, because it's contrary to your hypothesis, because if this cell line doesn't express these receptors, maybe it means that these receptors are not involved in cell migration of these cell lines.
That's true for the X cells, but the UID7 line naturally expresses both receptors.
I think that it would be
it'd be interesting. Yes.
A better model. And I had also a question regarding ATG5.
Yes.
ATG5 is able to interact with BCL2, and I was wondering if when you overexpressed ATG5, did you found any cell deaths, apoptotic deaths in your model?
We had none. That's also a question, that's a very good question that we wondered, since it seemed that the cleavage of ATG5 gives rise to that smaller fragment that I showed, and it's that smaller fragment that is able to translocate to the mitochondria. So we wondered if we might have more apoptotic activity, but we didn't find any when we overexpressed ATG5.